to give the membrane its fluidity so it won't solidify like butter. And this fluid mosaic model, just a description of the whole system, the lipid biolayer theory, that the proteins are in the membrane, and that's how the membrane is structured, and that sort of thing. And that whole thing basically is just called the fluid mosaic model. These things are floating around in here. Okay. It's no more complicated than that. One word you're going to hear in the future, especially if you go into microbiology and things like this, is this glycoprotein. Okay, glyco, like glycolysis, like glycogen, it refers to starches, sugars. Okay, you put a sugar on a protein and you get a glycoprotein, and that's supposed to be the little sugar chain right there. Okay, and so there's a very common, and the bacteria use them to protect themselves and that sort of thing. And it's you have antibiotics directed at these things, but they also identify different types of bacteria by the types of glycoproteins they have on their surfaces. So that's a word you're going to hear again. So it's not really terribly important for us right now. For what I do want to say, membrane proteins or transmembrane proteins and integral proteins, I sort of use those interchangeably. They mean the same thing. So you're going to hear that a lot. So why did I start out with that cholera story? Really, I was just trying to get y'all thinking about the fact that diseases are not just things out there. That it's not magic. It's like I said, measles is not just a person who has little red bumps all over them. Okay, there's something going on at the cellular level, and when you start thinking about medical problems, that's how you start thinking. What is the actual problem that's going on? And you can almost always describe it on a cellular level, what's happening. And so this is a cholera bed, and this is not a disease that's gone anymore. Anytime you have breakdown in hygiene and things like that, you can have cholera epidemics. All you need is a carrier to get the infection in the water. It's a bacterial infection, and it's still all over the world. It's just Pakistan and Haiti just had a big outbreak after the earthquake. But this is actually what's going on. It's not just they got diarrhea. They have a toxin in their system. The bacterial toxin is affecting the chloride pump in the intestinal lining and the epithelium. Okay? And it lets this chloride flow out into the intestine, and because you have to balance charges, the sodium is going to go with it, okay, to keep the charges neutral. And now what do you have? You've got your intestine fluid full of salt, okay? Where do you think your body fluids are going to go? Out. Right, your body fluids are now hypotonic, and they will flow just by osmosis into that salt solution. All right, so you're just going to poop that out, okay? And it's so much that they just throw them in a bed with a hole that goes into a bucket, okay? And you have thousands of people like this sometimes, all affected at the same time, and they will have so much diarrhea. It says up to 6 liters of water per day. Well, I've even heard it's more than that. That'll kill you in 24 hours, okay? And so you need to really get these people on IV fluids. Or they have something called the World Health Organization has a universal cholera formula for rehydration fluids, and they tell you exactly how much glucose and salt to put in here and to make these solutions, and you just get these patients to drink as much as you can to replace everything that's going out. Okay? That's the only way you're going to save many of their lives, because this disease is, I forget the exact numbers, but it's like 60% mortality. Okay? And you're lucky if you survive it. And it's especially bad on the old folks and young kids. And it doesn't take more than a liter or two from a child for it to die of dehydration. Okay, so this is really nasty stuff. But so it's not just diarrhea. It is a disease. It has a reason. There's a bacterial toxin that causes this. And this is what the cholera looks like. Bacteria, they're rod-shaped bacteria. They're a toxin. They secrete this out in their system. Out from the cell, and this is kind of tricky little biowarfare game they're playing with humans, because this particular part of the toxin clamps on to the cell membrane receptors in the intestinal cells, and they basically can't tell that it's an enemy. Your cells don't define it as an enemy, and they bring it in thinking it's food. They bring it in a vesicle, and then the lysosomes cleave this thing off right here, and now you've got this toxin floating around in the cell, and this is what grabs onto those chloride pumps and turns them on and just lets that chloride just flow out into the intestines. Okay? So it's a real tricky little biowarfare game that's playing here. But you can see why a lot of people are worried about real biowarfare, because all you got to do is put some other kind of toxin that might be even more deadly, maybe something that shuts down the ribosome or something that shuts down the mitochondria. Hook it up to this kind of thing right here, okay, and put the gene in bacteria and then spread it around. See what happens. You know, that's scary stuff, but that's what's going on out there. So again, I'm just going to go through a few membrane proteins. There may not be any sense to it right now, but we'll be talking about a lot of this stuff later as well. One of the most important ones you have is an active transport pump, okay? And that uses ATP, 1 ATP, to put sodium outside the cell and potassium inside. The exact number happens to be 3 sodiums to 2 potassiums in. You don't really have to memorize that, okay? Just realize that all life has this pump, okay? Every cell has this pump, microorganisms and us and everything, because this is how they keep their osmotic balances going, and so that the bacteria don't explode for osmotic pressures and things like that. But this is real important to us. All cells do this, and without it, there wouldn't be any possibility of muscle contraction or nerve conduction, because you think of your nerves sending an electrical signal down to your muscles to set them off. Well, it's only electrical in the sense that it's charged, because what is actually being sent down and moving along the membrane are sodium ions, okay? Because they're building up on the outside of the cell, okay? While potassium is building up on the inside of the cell. Okay? But if you have another membrane protein right here that can be turned off and on and let the sodium rush back in, you're getting positive charge rushing back in, okay? And that is a nerve transmission. That's what happens. It's the sodium ions that are moving down the membrane causing that transmission. So it's not like electricity where you have the electrons moving around, okay? So, but we'll talk a lot more about that when we talk about those excitable membranes, and we go over muscle and nerve function. So, but this is the basis of it, okay? And this is really what it looks like too. This is fantastic stuff to me, because when I was in school, all they could do was do this, okay? Now we can do that. We know what they look like, we know the structures, we even know every single amino acid in the chain, because you can find the genes in the DNA that makes it, that's coded for it. You can check for mutations and things like that. You're not going to have many people alive that have any real nasty mutations to their sodium-potassium pump. This is one of those things that is very conserved. The gene is very conserved the way it was millions of years ago, because if you change it much, it doesn't work, you die. Okay, so you're never born. So that's why genes like that stay the same for millions of years without many mutations. Okay, and that's how we know they're very old. Okay, but this is really interesting. It's got a few alpha helixes there and beta sheets down in here. The yellow ones are usually beta sheets. Okay. But there's a pore that goes through the center there, and we even know the amino acids that are lining the pore that have OH groups, or the carbonyl double up on oxygen that sort of hydrates that sodium charge as it goes down through the pore. Okay, because there's no water in there to hydrate the sodium like it is out in the water. So it's made perfectly just to let that sodium ion fit in there and move down. Okay, and then the potassium. Then the ATP pumps it all the way through. So, but they're cool little things here. Yeah, so, there's another pump that's pretty important for us, the gastric acid pump. It's a hydrogen potassium ATPase. Anything that's an active transport, sometimes they'll put ATPase on the end of it. The A's meaning it's a sort of an enzyme, but it's an enzyme that basically cracks ATP to do something else. The ATP is the active ingredient there. And your stomach has a lot of these, obviously. The parietal cells, don't worry about these names, but just it's a specialized epithelial cell, okay, in the gastric lining. Sometimes that can get a little too active. This is a shot from a gastroscopy. Here's the ulcer region right here. I'm sorry, it's a little blurry when you blow up the photo here, but this is the first part of the intestine, the duodenal area there. This is what they look like on the scopes. And it used to be thought that, this is for the PBS moment here, it used to be thought that most ulcers were caused by too much acid in the stomach, and just too much stress, and things like that. But that's kind of ridiculous because most people are under stress and most people don't get ulcers, right? It turns out, it was almost by mistake, there's some guys in the early 1990s that were scoping and looking at all these ulcers, and they were taking little biopsies, and they started to culture them. They just put them on those agar plates like you saw in the lab, where we were doing that little diffusion experiment. And it turns out they were, almost all these ulcers were growing the same kind of bacteria, something called Haemophilus pylori. And we call it H. pylori for short. And then they thought, well, is this bacteria causing the ulcers? And they were laughed at for, a year or two. But they proved it. They proved it, that most ulcers are caused by H. pylori now. And now you no longer cut out stomachs. That was the only way to treat these things. When they started bleeding, you went in and you did a gastrectomy. You cut out that part of the stomach and then sewed it back up. Thousands of people had this surgery done. Thousands, if not millions. A lot of people had ulcers. And now, when people come in complaining of stomach ache or heartburn or whatever, they actually do a blood test and check for antibodies against the H. pylori bacteria. Okay? And if you're positive, they treat you with antibiotics and Prilosec, the anti-acid medication. Usually within 2 or 3 weeks, you're cured. It's gone. And you never get the ulcer. It never gets that bad. Okay? So we don't see bleeding ulcers much anymore because of that reason. Okay? But we used to see them a lot. I remember at least 10, 15 cases when I was working in the hospital and in the ER, where people would come in throwing up blood and all sorts of stuff, and you had to put these big plastic tubes down in their stomach and flush it with ice water and give them transfusions to try to keep their pressure up, and it was tricky. You could lose them, and so but you just don't see that much anymore. Okay, so and if you're wondering, it's this pump that Prilosec and Zantac and a few of these other things, that's what they shut down. Okay? Prilosec will kill all the proton pumps in the stomach for about 3 days. Okay? And so your stomach is not producing much acid for that period of time until your normal processes of turning over the pumps. Your stomach lining is the genes there in the DNA. They're making that protein all the time. So until they make enough and enough more pumps come online, your stomach doesn't have any acid. Okay? Because it permanently shuts down the acid pumps that are working at that time when you take one of those pills. That's how it works. So here's an actual gastric ulcer. You can see why these things are dangerous, because all this is eating away at the stomach lining, and all it has to do is hit an artery. Okay? And then you are just going to be in trouble. Okay? But this is why pancreatitis is so dangerous too. The same thing happens when you get inflammation of the pancreas, or you get a gallstone that goes down the cystic duct and blocks the pancreatic duct because they go in the intestine in the same tube. Okay? You'll very often get the gallstone pancreatitis, and that pancreas now, all those digestive enzymes are digesting itself. Okay? And all they have to do is eat into an artery, and you're going to go. I've lost a couple patients from that. So, okay, so again, I'm just throwing out some membrane proteins. There's no rhyme or reason to them. This is aquaporins. You'll notice all the alpha helixes, and they're sort of arranged in a circular pattern, if you could see the whole thing. So there's actually sort of a little hole in the middle that water goes through. Okay? And there's only about 1 or 2 water molecules that will fit through that hole. It's not a big gaping hole. So this, and it's specific for water. There's not much else that gets through. And this is the type that mammals have. Okay? All these alpha helixes arranged like that. It's an integral protein again. The membrane, the top phospholipid would be about here, and the bottom one would be about here, and the tails would be in here. So you can guess what kind of side chains this part of the aquaporin has. These are the non-polar, hydrocarbon kind of ends. They're going to stay in the fat, whereas the outer part might have some water-soluble proteins to hold it, there. And this stuff is not, it's not old. It's like we're standing on the shoulders of recent people who have done good work to understand this stuff and how it works. Nobel Prize in 2003. Okay, so this is not like this was even, the structure certainly wasn't known when I was taking classes. Okay, so now we know. We know the kind of mutations that can affect it and cause them not to work very well too. So, and then this the potassium channel, the same way. That's a very important channel for nerve transmission and all sorts of things. And it's relatively new that we know how it works and we know what we can do to try to help patients with it. But this is one of the interesting things that I find. This is the porin for bacteria. Okay? It's just like an aquaporin in mammals, in us, in animals, but look, it's all beta sheets. No alpha helixes. Interesting. So we know that our history is quite different since we were all that one cell thing. But look at this. The porins, the beta sheet pores, are just like that we have in mitochondria and chloroplast. That's another reason we know that mitochondria are bacterial in origin. Okay? Because the mitochondria make their own porins. They have their own DNA, okay? And it's bacterial in origin. So, interesting stuff. And here's a shot of that potassium channel that I just mentioned. Big, important membrane protein. Virtually all living organisms have these things. Okay? Because, like I say, these sodium-potassium ions are so important to life that controlling their flow is necessary. Okay? The interesting thing that we have a couple of different types. We have channels that can be turned on by chemicals. We have channels that can be turned on by voltage, changes in voltage. And this potassium channel here is a voltage-sensitive channel. In other words, let's say this is open, and you can see the potassium ions coming through. Okay, they're going through by diffusion. This is not a pump. This is not an active pump. The potassium can be going through, can be leaking out usually. Okay, because there's more potassium inside the cell than there is out because of that other pump, the sodium potassium pump. So usually it's leaking out. Okay. Or it could be shut, but depending on the voltage underneath the membrane here, it'll either be open or shut. Okay? And that's how we control nerve impulses. Okay? It's the flow of ions that we're controlling when we control our nerves. And like I say, we'll go through that a lot more when we get to that section. But, this is just fantastic stuff. Here's another membrane protein. They don't really show it, but the membrane would look something like this. The phosphate head's about here and here. Okay, so this is what is sitting on the epithelial lining, or the membrane lining of the olfactory cell, the cell that makes these things. It has the gene to make this protein. This gives you your sense of smell. The interesting thing about smell is that you wouldn't think, well, you might, because smell is a very primitive kind of sense. Okay, if you have a receptor that is sort of designed through evolutionary forces, there are certain types of chemicals that fit in there, and that sends a signal inside the cell. Okay, so it's a sensory organ. Okay, very primitive. All it takes is an actual molecule to land on there and set it off. Okay, so that you can think that developed long before something like vision or hearing or whatever. So these are very ancient sort of proteins. Mammals actually have about 1,200 genes. It's the largest gene family in our genome, where there's about 23,000 genes out of all our 3 billion pairs of bases in our genome, in the DNA. The biggest family is 1,200. Did I say 12,000? 1,200. 1,200 genes that only code for one protein for smell. Okay? And The thing is, we've lost most of them. Humans now only have about 400 that work, okay? Because we don't live by our sense of smell anymore, okay? But animals like rats and other things and dogs, they do. They have a lot more of the same genes. It's all the same genes because we're all mammals, right? But they have 600 that work, and rats now have, I think they've counted 900 to 1,000 that are still working. So they have a much better sense of smell than we do. But we don't live by our sense of smell, so if one of them gets mutated, so what? It's not going to affect your life much. So what if you can't really appreciate the true smell of a banana? It doesn't really affect your life much. So, but it does to animals that live by their sense of smell. So the mutations are going to really hurt them as far as their reproductivity, but it doesn't us. So we're down to about 400 that work, although we still have all the others. We have the other 800 genes, but they're so mutated that we don't even turn them on. They're just there in our genome. But the way these work is this is just a site. You can imagine that only certain things would fit in here, certain types of molecules. Okay, and when you get one that fits, it actually causes a mechanical shift of the shape. Okay, and it's the movement of the tail down here that changes shape and allows other proteins to latch on when the shape is changed. And then they are enzymes that generate things like cyclic AMP, the one I told you about, the ATP making that little cycle. Okay, that's what they generate, and that's the signal that goes and turns on other membrane proteins that let sodium or calcium rush into the cell, and that's the electrical charge that goes to the brain and says, hey, we just smelled something. Okay, so it's not a direct opening like some of our proteins are, but that's how your vision works too, actually. It's the change in the shape from the light hitting the molecule that sends that signal to the brain. Okay, that's how it works. But the thing is, yeah.
When people lose their sense of smell, is that those die or what? What happens?
Normally, yeah. If you're talking about they had a good sense of smell and then they lost it,
Yeah, they just.
it has to be something pathologic. It has to be chemical. The coldies, the zinc cold tablets. The people that came up with that also came up with a nasal spray, and about 1,000 people reported that they had completely lost their sense of smell after using that. So they took it off the market. Can't get it anymore.
Wow.
So yeah, it's called anosmia, when you can't smell. This is the medical term. But yeah, they didn't report it for a long time, and there was a lot of lawsuits about that. But it's actually zinc that's in there. They think it's the zinc metal ion that blocks these things and kills them. So, but these things are pretty interesting the way they work too. PBS at the moment. We can actually differentiate. Now you know what fatty acids look like, and aldehydes. Usually they smell better than acids. Okay, fatty acids smell like fatty acids, like fat, butter, that kind of thing. But you take off that hydroxyl group and make it an aldehyde. Then they tend to smell pretty good. They smell like flowers and food and things like that. These are nice smelling things. We can tell the difference in smell between an aldehyde with 9 carbons and an aldehyde with 10 carbons.
Wow.
That's pretty cool. That's pretty cool. Because they fit in different sites in our nose. But the way it works, the smell is not in the molecule. We have 400 of these different types of things, and one molecule might actually fit in 5 or 6 or 20 different types of proteins. Okay? It's the combination that your brain recognizes as the smell of bananas. So there's really nothing about that molecule that smells like bananas. It's your brain that's telling you that's the illusion of smell. Okay? Your brain is telling you it smells like flowers or roses or whatever. But it's just the fact that these compounds are setting off different kinds of patterns in your olfactory epithelium, and then your brain interprets that as a certain smell. Okay? And we're wired to think some things smell good and some things smell bad. Okay? Nobody likes the smell of feces, right? Okay? You can't help it. You're born that way. You're wired for that to go into your sense, the area of your brain that senses that disgusting sort of smell. Okay, you can't teach yourself to like it, okay? And vice versa. Some good smells, we're wired to be attracted by them because usually it's food or something. Okay, and we're wired for that. So and you can't forget smells either. That's one thing. it's such an ancient sense that it goes right into your amygdala, right into your memory. You can't forget what your grandmother's house smelled like. You know, you can't forget what bananas smell like. You can't forget what apples smell like. You know? So it's a weird kind of thing. It's wired in your memory a lot more than your vision and hearing. So I find it a really interesting sense. So otherwise, it's just an example of a membrane protein. Okay. Mitochondrial ATP. I showed you that little wheel. These are all membrane proteins. Okay. All these globs here, and there's several sets. There's probably a couple of dozen proteins that make up these things. But what that does is give you the possibility that you can have a genetic mutation in the gene that codes for that protein, and you're going to get mitochondrial disease. If you can't use that oxygen very well, if you can't shuttle the electrons through the system very well, if you can't pump out the hydrogens that you need to run that pump, okay, any of those proteins, any of them, you screw up any of them with a genetic mutation, and you're going to have a problem if you live at all, okay? Because obviously if the system doesn't work, you're going to be miscarried. You're never going to be born, okay? So, but if it almost doesn't work, you can be born and you're going to have a disease of mitochondria, which is usually going to affect your nerves, your muscles, and most of your other cells too, but you're really going to notice it in the muscles because you need so much ATP to run your muscles. So a lot of the symptoms of mitochondrial disease are muscular problems, weakness, double vision because your eyes can't track together like they should. Your eyes are really sensitive muscles, okay? And there's probably the best controlled muscles in your whole body, okay? Because you have to keep them tracked together and focus on one area. And any little lag of any muscle on any side, you're going to see double. Okay? So when you have mitochondrial disease, double vision is one of the big complaints. Okay? Yeah.
So is that when a person is cross-eyed, is that a malfunction?
No. That's usually a muscle or nerve disorder.
Oh, okay.
Yeah. It's much more rare to see mitochondrial causes for that.
Oh,
okay. I'm talking about not a permanent dislocation of one of the eyes. You know, I'm talking about the fact that the muscles are just kind of weak and you can't control them well enough to track together.
Oh.
But it's not like you've got cross eyes. Okay.
What about a wandering eye?
That's the same kind of thing. That's usually a nerve disorder, not a mitochondrial.
I have a friend whose daughter, her eyes always like move around.
Nystagmus, always jerking.
They're always moving.
Yeah. Yeah. Well, I mean, I don't know her history, so I can't really tell you exactly.
Born like that.
She's born like that? Yeah, probably some sort of ophthalmic nerve plexus disorder. But the nystagmus is the name for eyes that jiggle. Okay? And you do that slightly all the time anyway, but you do it to such a slight extent that your brain doesn't appreciate the movement. Okay? And toxins, drugs will usually give you a vertical nystagmus. Your eyes will go up like that. Okay? Sideways nystagmus is usually more brain disease and things like that, cerebellar disease. So, one way you can use nystagmus to check for people that are faking blindness. Every now and then you get people that come in and want disability because they can't see, or it's just a hysterical kind of thing. Blindness is, all you got to do is take a strip of paper that we had them in the clinic all the time. There's always one in the lab somewhere. And if you have a patient like this, you pull out this strip of paper and it's got black and white stripes on it. All you do is move it in front of their eyes like that, and they can't help it. So they can see. You got somebody really blind, their eyes aren't going to move at all. So
Doc, what we're looking at, is this like a vague, diagram of what's happening in the mitochondria?
Yeah, very vague. Yeah.
Okay.
Yeah, but
okay.
this is the basics.
Okay.
This is what keeps you alive.
Right.
I mean, this is where you get your ATP right there from that paddle wheel. Okay. Let me just make this real clear, and I think this is the concept that I want to get to you eventually about what is a mitochondria. What does it do? The reason you breathe is to get oxygen right here to this protein. Your body really doesn't use it for much else. Okay? The peroxisomes use it to make hydrogen peroxide and detoxify things a little bit, but to keep you alive, the oxygen has to get here and here. Okay? That's why you breathe. This Krebs cycle here, this is where the glucose comes in. Okay? It's broken down, and eventually the parts and pieces will get into this cycle, and it will be broken down into carbon dioxide. So the carbon dioxide will be diffusing out on its own. Okay? Or it will be turned into sodium bicarb. Okay? As part of the buffer that goes around into the body. So this is why you eat. Okay? And this is why you breathe. It's all mitochondria that are keeping you alive. Okay? And the only reason you do that, okay, is so that you can shove hydrogen out in here in this space. Okay? This is the inside of the mitochondria. This is the actual wall of the mitochondria, but this is a space that's, you know, what's the word? Sequestered on its own. Okay? So those spaces aren't connected. This is a space in between. And the only reason that you're doing all this is to pump hydrogen out here to create an acid environment. There, it is about 4.5 here, pH, because it's got hydrogen, it's more acid. Well, now that acid wants to go somewhere because it's getting concentrated, so it wants to diffuse somewhere. Okay? And then the mitochondrion says, ha, you can get back in, but you're going to have to run this paddle wheel to get back in. Okay? So it's like it's running down a concentration gradient, just like water would be turning a water paddle wheel. Okay? And that gives you the mechanical force of that protein that's squeezing those ADPs and phosphate to make ATP. Okay? And then these are taken out with their channel proteins that carry the ATP out. Okay? So there's more openings in here for the ATP to get out. There's openings for, you know, the sugars and things to get in. So this is kind of simplified. But this is basically the secret of life right here.
And when we look at like a picture of the mitochondrial, you see all those like folds, and it's like that in one of those areas, and then that's another one, and another one, another one, and all those folds.
Those folds are called crista.
Yes.
Okay.
Yes.
And this is why the folds are there.
Okay.
It's those membranes. Okay. And I'll show you some electron micrographs of that coming up. So what do I want you to know? You don't have to learn all this stuff like I did, all the names of these proteins and blah blah blah. Okay. Just have that concept of, you know, the reason I breathe, okay, the reason I eat, other than to build me, okay, I need energy, I need ATP, and the mitochondria is doing that for me, okay, and that hydrogen in there, this is one of the few places in the body, besides the lysosomes that use acids, okay, that will have a acid pH, okay, in the cell, okay. And that is only because we want to run that through this pump here and spin that thing around and make ATP. Okay? So that's the concept I think I want to get across. And I already showed you the video, so this is kind of anticlimactic here. But again, the hydrogen coming through, you see how many proteins are invested in this structure. Every one of these things can be made wrong. You have a genetic defect in the gene that makes that protein, you're going to have mitochondrial disease if you're not born dead. Okay? So and there's the protons in there. They go in, and the only way they can get out is to turn this thing. And it's all just electrostatic forces. Okay? The positive charge is being repelled or attracted by something in that protein, which makes it turn. Okay? So it's just like I had a south end of a magnet here and a north end over here, and I put something there, I can make it turn. Okay? It's just an electrostatic kind of thing as it goes around. It's a fantastic little machine. This is pure PBS. If you want to shut down that process, you want to kill your mitochondria, you can use rotenone. Right? Rotenone is actually, it was found by South American natives, and they use the roots of these plants. They grind them up and make this milky kind of mush out of them, and then they just throw that. They go out on their little boats and they just throw it out in the water and let it sink down. It's just like throwing, you know, it looks like flour when it's out in the water, just sinking down. And a few minutes later, bubble the fish, because this is a mitochondrial poison. Okay? It shuts down that electron system. Okay? Interesting little molecule. Part of this molecule right here, NAD, has a structure that's very similar. It's very similar to the quinones, like this. Okay, and I have a feeling that this is the active part right here, because this probably gets reduced to an alcohol and it shuts down the transport. Okay, it grabs the hydrogen out of the system, the electron out of the system, and just shuts it down. Okay, so because these are pretty active, especially when you put ketones next to double bonds like that. So, here's an oxygen-carbon, oxygen-carbon. We didn't really require you to know that one, but those are ethers. Okay, that's an ether. That's an ether. This is a ketone. These are ethers here: carbon, oxygen, carbon. So these are all ether linkages, but they're all functional groups. It wouldn't be too hard to make that one up. A lot easier to go down and get the plant. Okay. All right, now let's say a few things about the organelles as a whole. Okay, so we're going to talk about just the nucleus a little bit here. You remember when I told you when we were talking about prophase, and we said the one thing I want you to remember is that the centrioles are separating and the chromosomes are forming and the nuclear envelope is dissolving. Okay? Well, when you say stuff like that, it's just not to confuse you later on at the time, but there's really no such thing as dissolving nuclear envelopes. And you could go, what? How does that happen? You know, what are you talking about, dissolving? Well, we found that the nuclear envelope is a double lipid membrane. It has two of those lipid bilayers. Okay? And the outer one is continuous. It folds into the endoplasmic reticulum. Okay? So when you want that nuclear envelope to dissolve, all you do is just fold it back into the endoplasmic reticulum. It's all membrane. Okay? And then when the chromosomes come back together again in telophase, you can just unfold that envelope again around them. Okay? That's what happens. That's what happens. Fascinating. The nuclear envelope also has a real special system of pores here. And these are not like the aquaporins that let water through. These are huge pores with dozens and dozens of proteins separately hooked together to let in big molecules and let out molecules like RNA. Okay? You're gonna decode your DNA. You've got this huge RNA molecule that has to get out. Okay? So it has this weird pore that looks like a sink drain in your sink. Okay? And it actually has little prongs coming across that only lets the RNA go through in a special way and twist out. And so it's really a fantastic little design too. But that's what all these things are. Okay? And then the nucleolus here, remember this is making the RNA for ribosomes, because the ribosomes, which we'll see in the future here, are mostly RNA. They're an enzyme, but they're made mostly of RNA, not protein. Okay? They're about 30-60 or 30-70 of that for the protein RNA. So they're a real special organelle. But the nucleolus is what's making that special kind of RNA for the ribosomes. Okay? Okay. So, and here's sort of a cartoon drawing of what it looks like. Okay? Here's, you can see your double membrane. Here's your lipid bilayer folding over on itself like this. And this membrane, or pore here, is a huge set of proteins in there. This is real simplified the way they do, but it kind of acts like this. And you got to go around these things to get in and get out. So here's chromatin, and here's chromatin along the edge of the nucleus. And they found that these things tend to pack like this next to the pores, probably because when you're making the RNA, you can somehow guide it with other proteins and just slip it right through the pore and get it out into the cytoplasm. Okay? So making things out here would be more difficult to find their way in there. So this chromatin can be, like I said, be decoded and then send the RNA right through the pore. Here's one pore, here's the other. Okay? And this
are those pores called annulus? Is that what those are called now? You had it written on, go back one. You had what is that on the bottom?
Down the bottom, A-N-N-U-L-U-S.
Is that what those are called?
See him? Yeah, right
The
there.
annulus, the ring. Annulus to me means circle. And these things, I don't know, I think that's a separate pore. Each one. But I guess you could have pores arranged or something. I'm not sure. I'm not really sure about that terminology. Sorry?
It's not gonna be a.
No. I'll be happy if you just remember that nuclear membranes have pores. Okay. All right. I'm just trying to get you excited about it a little bit here, okay? But this is what they look like from the top, okay? And it's, you can see they really do look like a sink drain, okay? And on the bottom, you can see the strainer. Okay? This is looking at the outside of the nuclear pore, and now you're inside the cell, or inside the nucleus, looking up, looking out. Okay? Fantastic little structure there. But that's to keep other things. That's to keep other things from getting in the nucleus and screwing up the program. Okay? But obviously, other molecules do get in. Okay? So it's not perfect. The reason that many chemicals and compounds are carcinogenic is because they do get into the nucleus and put a peg in your DNA and cause mutations.